Our next speaker for today is Professor Mitali Mukerji from the Indian Institute of Technology at Jodhpur. Welcome. So Mitali has obtained her doctoral degree from the Indian Institute of Science at Bangalore. And she was, for many years, the chief scientist at the CSIR Institute for Genomics and Integrative Biology. Now she heads the Department of Biosciences and Bioengineering at IIT Jodhpur. And she's a molecular geneticist. So she works on understanding the human genome structure and what it can say for how we deal with, how we are able to predict, discover various biomarkers. Her work has earned her a lot of recognition. She won the CSIR Young Scientist Award, and she's also a recipient of the prestigious Shanti Swarup Bhatnagar Award. So today she is going to tell us about this field which she's been working on in the last several years, Ayurgenomics, a conversation between Ayurveda and modern medicine.
Good evening, everybody. It's really humbling to, so I would like to thank IMSC to give me an opportunity to speak in front of such a elite audience, and I hope that I do justice to the talk because this is a very unusual audience for me, and the topic is also a little different. So we'll get on with it. So this is IIT Jodhpur. It's a beautiful campus. I just moved 1.5 years back, but from IGIB, and it's a very relatively very secluded, very beautiful place. So in this place, we have just set up a Center of Excellence in Ayurtech, and I will towards the end tell you what we've been doing and why we've been doing these things. Okay, so so a little bit of my personal health journey, and you might be curious why you would like to want to know about my health journey. How does it matter to hear? But nevertheless, I'll relate this story and let's see whether we can make anything out of it. So till the 30s, when I was younger, I lived in a relatively small townships and all. Stresses were very less, and only thing was there were frequent displacements, but they were all in small places, and nothing much happened except that I really had these severe sweating, which many of us as kids might be having. It's a little uncomfortable, but you don't go to a doctor because you don't know what you tell the doctor, right? And then there were acne, which also happens to be there in many people, and lots of people give lots of recommendations. Grandmother gives lots of suggestions, and the doctors load you with lots of antibiotics, and that's pretty much what you get to do. Then came the after 30s, where we go through careers and stresses, and I happened to move to Delhi to establish an institute. So, so you are having different kinds of exposure than where you've grown, and there's again an urban lifestyle. And here there were some problems happened to me. So I had a bleed in the brain, so which required a surgery, and then the other kinds of things where you gain a lot of weight, or you have diabetes, or you have some other kinds of complications which are, some are treatable, some are not treatable. And this is the time when I also delved on this subject, because I've been working in this for the last 2.5 decades. And here some of the things I could work it out with my surgeons who treated me and I walked back to life, or there were Ayurveda doctors with whom I was working. But one very interesting thing happened, a chance talk with the Ayurveda doctor whom I was collaborating with, and a 15 days of medication, and I was totally off my sweating. So something which had totally, my quality of life was very uncomfortable. If you hold something, it becomes wet. If you wear a shoes, it becomes wet. And we didn't know that there was something to be done, but this 15 days of Ayurveda medication totally cured me. I don't remember ever having sweating. Now I have moved again to a small city. Lots of stresses are gone. having one retirement and a newer job. But I've gone to a stage where I've aged a little bit, and it's also a very desert environment where I've gone. It's a relatively different environment than what my native roots are. It's a very extreme, harsh environment, and I continue to have diabetes control. But I also had mild COVID in these times, but the COVID was not so, meaning not so severe as other people had. And again, some aging related issues. So now what do I do? So there are, during COVID time, I tended to, because I was working in Ayurveda, I tended to use it during prevention time, during treatment time, and really, I really do not know how do I further, how my trajectory is likely to be, or how do I move forward. So now, whatever the story I told you, as many people in the audience might have gone through certain health trajectories, everybody would have some stories to tell. And if you aggregate all these kinds of information, turns out that there are certain patterns coming up. With the global health disease scenario, longevity overall has increased in all parts of the world. We've been able to battle lots of infectious disease and all. But what has been shown by studies across many diseases, around more than 350 diseases across different countries, is that our health longevity has reduced. So as a result of which, we are living for a longer time, but we are not, the last decade of our life, we do not live in a healthy manner. And majority of this is related to non-communicable diseases, which cause substantial economic burden. So the need is how you can increase the health longevity. You live longer, but you live longer with health, and that's what is the need of the hour. So in this context, this area which is my expertise and which I've been working for the last, again, 2.5 decades is genomics. And why is genomics? So genomics is something which I'll come to, but this is an area which actually promises that you might be able to predict what you are predisposed to so that you can prevent, you can participate in your health management, and you can also have a personalized approach to how you manage your disease. And this is where it comes from womb, how you plan a baby, when you plan a baby, to right till the cause of death. So, so there are lots of screenings one can do to prevent fetus born for some abnormality. You can do some preventions when a newborn is born with certain kinds of defects. You can also maybe diagnose a disease before it sets in, and you can combat certain diseases like infectious disease, cancer, or even if you are having medicine, some people have side effects to some medicine, you might be able to predict who are likely to have what kind of reaction, and also ultimately how you can increase a health span, or if people die, how do they die, what do they die of? Those are also important questions to know. So, so little bit about, just to get the entire audience with me for the subsequent things. So what is this basically genomics all about? It's basically a book of life. It's book of our life written in our cells, in our chromosomes, which have the DNA. The DNA is our genetic material. And this is a chemical letter, DNA discovered, or the structure was elucidated by Watson and Crick, as many of you would know. So just to kind of equate with what we would mean by why we would call it as a book of life, it is like we can equate DNA to letters, and if you thread these letters, you have words, which in the genetic or in the molecular language you call as codons. And then when you stitch these codons together, or when you string these words together, you get sentences, and which are equated to genes. And if you have these genes put together one after the other, they make something which is the chromosome, which is like a chapter. The sentences form chapter, and then you put all the chromosomes together in a cell that is a genome or a book which you inherit from your parents, one book from one father and the other from the mother. So the entire how you will be, how you look, what you are predisposed to are all written in these letters, and these letters are around 3 into 10 to the power 9 base pairs. So how do these simple chemical letters turn to become these, give these functions? Is just because these genes could open up and transcribe to some things which is called as a messenger. This is basically called the mRNA. And this messenger is then read by the machinery, cellular machinery, and it recognizes these letters in triplets, and it starts incorporating amino acids together. And when the whole gene is read, it becomes a protein by combinations of these amino acids. So we have 20 amino acids and we have so many mRNA, so you can well think how the combinatorials will give rise to different kinds of genes, and these could encode different kinds of proteins. So the human genome has around 20,000 or odd number of genes. Numbers are still debatable, but these basically do not express in all the cells every time. Otherwise, we would have all the same kinds of tissues. The fact that we have hand, feet, leg, and all those are because some of the genes or some of the are read in some cells, some are read in some cells, as a result of which you get a whole human body. So now you can imagine that if there are changes, because this has to keep on replicating, the cells have to keep on dividing, so there could be errors as you are writing something and you can type and you can have an error. Similarly, when it copies itself, it can make errors, and when it makes errors, it will make some errors in the RNA, and then that can make some errors in the codon, and that can incorporate a wrong amino acid, and you might get a wrong protein or a different protein, and that's how you get so many differences. So long back in 80s, people thought, why don't we read the book of life entirely? And if we know a kind of a reference book, then we can compare it with different humans across different continents, and we can start to make sense out of how we are. But it's been a very humbling exercise. Technologies have expanded and we've been able to read millions of genomes, and now we find that there are too many variations, and we really do not have a definition of what is a healthy or what is a reference human genome. So from the genome, one cannot say who is healthy or how do you define health. And now there are other kinds of challenges coming up, that there are no reference genome. And in the practice of medicine also, people are finding that whatever drugs people do a randomized controlled trial, and when they really administer it to people, there is a very variable outcome out of the same medication. You would also realize in the same house, if somebody is hypertensive, everybody does not have the same medicine. Not only the same medicine, not only at the same time also. And that's because every of these trials have been done on populations, and populations have inherent variability, and as a result of which now these figures are coming out, and these are outcomes of large number of trials where you see these number of red colors. In each of these are different unfortunately the pointer is not working, but if you see these different panels, there are individuals who are in red and one in blue, and this basically just tells that if you have that medicine, how many people have an effective outcome, and this is given by a measure which is called as number needed to treat. So typically out of 5, if there is one blue, that means if 5 people are given treatment, only one has an effective outcome, so the NNT is 5. Or if you have something which is 50 and one is effective outcome, the NNT is 50. So as you can see that so many people are having medication, but if there is so much of variability, as a result of which one needs to now think through why in everybody all the medicines do not work, and that's why there is a paradigm shift that we need to move away from this kind of a population-based averages to something which is an individualized base trial. The other focus is on wellness. Now people have become much, much more aware, and more than treating diseases, as even more $1 trillion market, which is growing every year 5 to 10% on wellness. People want to be well, and there are too many recommendations, and people really do not know what they need to have, and they keep on having many, many different recommendations. The other aspects is, as I was mentioning, there is this another thing which is called as a precision medicine, that is from the population, how you can individualize and give a very precise medicine to a person at a precise time. That is, an individual is a moving target, the individual is evolving, the disease is evolving, the person is moving, and how you can start to map the person in all these spatio-temporal dimension and say that how you will calibrate your health, right? So that for that, people in US and other parts of the world also have started these big data collection of you have these active watches, how much exposures you have, how much water you drink, how much you move, and then your body's cellular readouts by way of your genome, your transcriptome, and they are trying to analyze them and try to see whether there are patterns which can tell from these patterns who are the people who are responding to a particular environment, responding to a particular disease, and on the basis of that, could you actually predict some signatures which will inform you beforehand, or will be able to allow you to define a targeted intervention. So this is what precision medicine is doing, collecting huge data on millions of individuals and then trying to work out what is the most optimum thing by clustering and doing all those kinds of analysis. Now, such a kind of thing about inter-individual variability, about precision medicine, has been actually described in India many, many years back. And this has also been acknowledged by people in the West. They started to say that in India, the concept of Ayurvedic medicines are first laid out in sacred texts, and the goal of the medicine is to maintain body, balance between body, mind, and then they call as spirit. And this is tailored to each person's something which they call as constitution or Prakriti. Now, there is a shlok in Ayurveda, which is from CSU, means charak, and Sutrasthan means the place where all the principles are documented. And if somebody can read Sanskrit, then the whole translation is maintenance of health in healthy and disease in diseased. So you can imagine thousands of years back, they have actually talked in a personalized context and not in a population context, and that's the primary aim of Ayurveda. Now, one can appreciate this much more as we have battled the COVID pandemic. Nobody was spared. People from any country, any economic background, any gender, everybody was affected, and each had a very different story. And modern medicine evolved in a very quick time by mostly trial and error. Something was in, then we hear that medicine is not going to work, something is there. And for every patient family who had lots of people affected, there was a ticking bomb. Am I going to need oxygen? Am I going to get into a crisis? Will I have an inflammation? Will I lose sense of smell? Everything was uncertain. And this was the time when people started also taking anything which was available as an information to avail medicines. Now, this was for the modern medicine the most challenging time. But for Ayurveda, it was actually business as usual, because for Ayurveda medicine, people really have to look at modern diagnosis, modern tests, and then they have to go back to their literature and try to evolve a therapy or a mechanism, and how do they do it? Why is it that for them, COVID was what they called as Jan Pad Udhans? That means a whole community is affected. And they actually had come out with, the Charak has actually described when does such kind of pandemic happen. And then there are descriptions around what to do in these kinds of pandemic. So it talks about the factors which are common for all the inhabitants of a country: air, water, location, and season, can actually lead to such kind of Janpad Udhvans, right? And during the COVID time, this is a small survey I also had circulated this. Many people availed Ayurveda medicine either by way of a treatment or by way of prevention, some with some educated knowledge and some with, as they were taking modern medicine, they were also taking Ayurveda medicine. So now I put the context. I just want to now get to why this Prakriti seems to be scientific, why it has stood the test of time, and what is this concept of individuality. So according to Ayurveda, an individual's constitution type, which is called as Prakriti, is fixed at the time of birth and it remains invariant all throughout their life. And all these, I am not a Pandit of Sanskrit, I am neither an Ayurveda doctor, and I have worked very closely with Ayurveda doctors, and these are all curated literature from clinicians of Ayurveda. So anybody who follows Sanskrit would also be. So many people ask us that if we say sentences which sound like precision medicine of today, is it really true and was it documented? So we actually have been having this practice of documenting a correlated term or a correlated sentence. So why, what is this Prakriti? So there is a common organizing principle, just like we had RNA or a DNA as a common organizing principle of living systems in the modern context. In Ayurveda, Tridosha is something which is called as a common organizing principle. So now what is Tridosha? There are three components in the body. One of them is the component which governs movement. There is another component which governs building up, and the third component is the one which governs metabolism. So there is a kinetic, there is a potential, and there is a structural component. And these are called as Vata, Pitta, and Kapha. Now every individual will have all these components because you need to build things, you need to break things, you need to move things. But they would be in varying proportion, and each of these do different functions which are described in Ayurveda. So these are just English translations of this. For example, manifestation of a shape, proliferation of a cell, or movement, signaling, transport, all the things which are kinetic in nature are governed by Vata. All the things which have a heat component, digestion, thermoregulation, immune response, these are the kind of things which are functions of Pitta, and the growth, the structure, the rigidity, or whatever is the potential component is governed by Kapha. So in an individual, you would have all the three, but if these three vary in proportion, then you are described as a person having an elevation of one or the other doshas, and as a result of which Ayurveda would describe people as Vata, Pitta, Kapha, or Vata, Pitta, Pitta, Kapha. Like that, they have 7 broad groups. Now, what is very interesting is, once you know these proportions in the body, then what is the disease state is called as the Vikriti. So Prakriti is your homeostatic state, and Vikriti is your perturbation from the homeostatic state. But each Vikriti is different for another person. For example, if I have more Pitta in the system, having more Pitta is my Prakriti state. But for a Vata Prakriti, having that much of Pitta in them will be a Vikriti state. So the purpose of Ayurveda is to assign what is your baseline health state, and then look at how you have got deranged from your baseline state and get them back to your baseline state, and not everybody to the same baseline state. And that is what comprises the way they would treat a person in a personalized manner. And all these things, the concept of the, dosha is evident in an individual. It is also used to classify a medicine, that what kind of dosha it balances, and it is also used to determine a cause or a trigger. So if a heat can elevate Pitta, so the cause is heat and the feature is Pitta, and then so you will have to have such a kind of a recommendation which will take down your Pitta by either elevating Kapha, so you cool the body and you reduce the heat. So this all thing about the interconnectedness of the feature and the causes through the therapy is what is the translational aspect of Tridosha, and this is called as Trisutra. So this forms the, today's day we are talking about network medicine, because we are trying to look at causes, features, and therapy in an interconnected manner, because that's what is now coming to this systems perspective of disease. Now this sounds abstract, Vata, Pitta, Kapha, signaling. So how do they assess how much is a person Vata or Pitta or Kapha? So it turns out that they actually measure a full system attribute right from somatotype, like how you look, your body form, to your skin type, to your physiology, to your taste perception, to your ability to resist a particular weather. So all, or your sleep patterns, and all these things are assessed, and then only people are classified into these constitution types. Now, these are the features of predominant Vata, Pitta and Kapha in terms of how they will look. So, typically a Vata person would have a more kinetic component. They will walk fast, they will talk fast, they will remember fast, they will sleep fast, they will wake up fast. So, there will be something which is like a movement. So, in Ayurveda, they call it as a chal component, or they have a laghu. So, they have a small body frame, small size, something like that. So, there are lots of descriptions based on which and a person can assess a person's Prakriti, and that would tell what doshas are more prevalent, and that would then become a part of their personalized regime. So, according to Ayurveda, any population can be stratified into 7 broad types, and once you have ascribed this form, then they can predict the trajectory, they can predict the function, and they can predict what is the regime one has to follow. Now, not only that, it's not static. Like, it's not that one time you have done a Prakriti. I was young, and as I showed you, my trajectory changed. I was lean, maybe 40 kgs, and then I became maybe 70 kgs. So now, if Prakriti doesn't change, and if I go to Ayurveda doctor, they will keep on telling I am a particular Prakriti type. But how do they do that? So there's very interesting spatio-temporal component in Prakriti also, and this is this is the only shlok which I really sort of understand, because it connects to the subject which I study. So they say Prakriti of a person is governed at the time of fertilization through the gametes, which is called as Shukra Shonit Sanyoge. Then they talk about the prenatal environment, mother's diet, mother's season of conception, so Matur, Ahar, Vihar, they talk about Kal Garbhashay Prakriti. So this is what leads to a person having a particular Prakriti. But not only that, it is impacted by ethnicity, the geography of the place, the season in which the person comes and presents to you, or the time. So they have Jati Prasakta, Kul Prasakta, Desh Anupatini. Anupatini means apportionment to the geography, Vai Anupatini, Kala Anupatini. And then you tell Praktatmyanita, that is the individuality. So when you look at an individual, they are actually contextualized in all these dimensions. And as I was telling you earlier, modern precision medicine is actually looking for this. And there is already a well-documented way of assessing it. Not only that, what are these Prakriti types predisposed to? So there are vivid descriptions about 80 diseases if Vata is more, 20 diseases if 40 diseases if Pitta is more, and 20 diseases if Vata is more. So this actually becomes a very, very good way to kind of have a principle as to how an individual health can be assessed. And what is very interesting is now people talk, COVID has come now, Ayurveda texts are thousands of years old. How will you evolve a medicine? So they have a term called yukti, and this is basically, it has all the reasoning way to how you arrive at a rational therapy by looking at the time, the season, the presentation and all. So that is how Ayurveda, it is a business as usual when COVID came. Now, so Ayurveda and modern medicine are very contrasting. Where modern medicine is very reactive, it targets the symptoms. Ayurveda is more proactive, and it actually targets the doshic imbalance. Ayurveda treats by monoing molecules. You're treating diabetes, you look at glycosylated hemoglobin. Here the aim is at reversal, more system observation. And then you know the nature of the molecules, specific molecules, you are having metformin. Here it is a polypharmacology. And then, but it is holistic, it is individualized, it is based on a person's Prakriti. So these are looking two different ways, and but you can think that why we would need to maybe have an understanding and maybe build a bridge so that we can get the best of the both worlds, objectivity with some systems perspective. So this is why it is needed, because if we have a whole lot of people with lots of heterogeneity, we need to break this heterogeneity into reasonable ways by which they can be treated more effectively, and maybe we can reduce the NNT and also increase NNH, which is number needed to harm. So we treat more people and we harm less people. But how do you get to that? So that's where I started my journey 20 years back. That is it possible to do that? I just heard about this and I was totally not oriented, knew nothing about Ayurveda. And at IGIB, we were doing genomics. We were trying to get prepared for precision medicine. And then we got to know about Ayurveda. And we asked a question, can we integrate these two sciences by the method of molecules of DNA? And that is where the Ayurgenomics came up, and that's been my journney. Now the challenge was, how do you build this? How do you integrate it, and when will you know whether it is integrated? So I just, this book of Jhumpa Lahiri, it's very, very interesting. She writes so vividly that you can visualize what she's written. Now one time in her life, she decided that she will learn a language and she will write her autobiography of learning that language in that language, and somebody else will translate it. Believe me, at least when I read the book, I felt as if it was Jhumpa Lahiri writing, because she had got under the skin of the language and she didn't do the English translation. Whatever she wrote in Italian was somebody else translated, and it actually read like Jhumpa Lahiri's book. So if we think about integrating Ayurveda with modern medicine or with modern sciences, we need to have a language which, if I read in molecular, Ayurveda doctor is able to connect, and if Ayurveda I take some hypothesis, I am able to query. So how do you build this language? So this is where we started a working hypothesis. This is a very, very old slide made in some version, ancient version. So I just kept it for historical sake. So I had a working hypothesis that if individuals are classified at the end of the spectrum as Vata, Pitta and Kapha, and they talk about signaling, metabolism and all, which we can measure. So if I take the peripheral blood of these people, would I be able to see differences, and will those differences have some interpretability in terms of Ayurveda description? So what we did, we took thousands of people. We were not getting funded in India, so we got it as an Indo-Russian project because Russians were also interested in Ayurveda. So we could do it in India, but it came with a rider that we had to go to Russia also and do Prakriti. But the thing was, nevertheless, we started, and we had Bhavna Prasha. She's an MD in Ayurveda. We worked together for over 20 years. So the idea was, if we can actually take populations and classify them on the basis of Prakriti and do all the state-of-the-art molecular analysis and try to synthesize the information. So we looked at RNA, we looked at DNA, we looked at biochemistry, and we started to synthesize and ask a question that how was it done. So when we did this, and we also wanted to assure that ourselves that we stick to one endogamous population, otherwise people will say that you took a Vata from north and Kapha from south and something from west, and then you are saying they are different, but they are just maybe genetically different because they come from different places. So I was also involved in making a genetic landscape of India in the Indian Genome Variation Consortia. So we knew that if we took people from here, they are more or less genetically homogeneous. So we took these individuals and classified them using a Ayurveda questionnaire into these three groups and asked a question, how do they differ? So when we did that, we found something very interesting. Anytime we do a biochemistry, we get a range that this to this, all of us are healthy. And if we cross that range, then we become diseased. But we found that Vata, Pitta and Kapha actually for many biochemical parameters tend to remain in one or the other range. So what would be true for one particular parameter would not be true for the other parameter. And that goes on to say that for Vata, the health state is a range within a range, and any movement in any other dimension will be a disease state. And this was something very interesting, because that means many people might be actually in a pre-disease state, and by the time they reach, they are actually in this maybe second stage of the disease, whereas for some, they will tip over to the disease state very fast. What we also found was, because we did expression analysis and we could bin the genes into what kind of functions they do, we saw some very interesting differences with respect to the kind of processes which were enriched in particular Prakriti. One such was cell cycle, which was elevated in Vata. And there is actually a shloka in Ayurveda. Vata regulates cell division and cell proliferation. It's called Parmanu Bhed Na Parisankhya. That's a work of Vayu, which was very interesting, and we have that in our supplementary, that this is what we found, and this is then the Ayurveda doctor could actually uncover that this makes sense, that if we are classifying, that means that it is possible to query Ayurveda through molecular means. So this was our first paper, and we got very, meaning we thought that, so now there is a way by which we can actually test out other kinds of things in Ayurveda. So then we went a little further and we said that if people say Ayurveda, Prakriti are predisposed to different kinds of diseases, do we actually see those differences, or is it just one way of classifying people and again seeing a difference? So then we started looking at genetic differences, and we found that there is an oxygen sensor gene. It was one amongst the 250 genes which differed, but we decided to follow up this gene, mostly for the reason that this gene was very understudied, so people would not have said that I picked up a gene and was studying it, because then again it will be one of, again, same study. So this oxygen sensor gene is very interesting. If we are in low oxygen condition, we need to calibrate our body so that we are able to cope up with low oxygen. Now this becomes very relevant if you go to high altitude and you have low oxygen. Some people do well and some people do not do well. Now we found in the sea level people of Vat, Pitta and Kapha, there was a Prakriti type, which is Pitta Prakriti, whose expression suggested that they would be much better adapted to high altitude. And the Kapha Prakriti type it was suggested that they might not cope up with low oxygen condition. So what did we do? We looked at natives of high altitude from our genetic data, and we found that actually Pitta Prakriti individual genotype is similar to populations which reside in Leh and Ladakh. So that got us interested, and then we asked a question. So then in that case, people go to high altitude and have this disease called high altitude pulmonary edema. Will they be more like Kapha type? And that is what we discovered. This was really a aha moment for us, and because it showed that even at sea level, you can classify people on basis of Prakriti. You might be able to assess who are the people who are likely to do well at high altitude. Then we again went to the descriptions of high altitude. Ayurveda, as I told you, described Desh also. So it had a description that Ayurveda, high or very high altitude, they call Kshitidhar, very Mahaparvat. Those places are very rich in Kapha. So if a person has already got Kapha, it will get elevated over there. So as a result, Pitta Prakriti might be much more adapted at high altitude. So this is, we published in PNAS for the first time. We could hit a big ticket journal because Indians would start recognizing me, okay, after 10 years, there's some merit in actually getting to work in this area. Not only this, after 10 years down the line, this has been replicated in multiple altitudes of the world. And people have also found that variability in this is linked to aerobic capacity, which is very important in exercise and all. So it tells us that everybody might not be suited for the same kind and same extent of exercise, and maybe some people might be better at some things. So this brings me to the operational framework. That is, I said Tridosha. Tridosha has to translate, right, to Trisutra. So we got Pitta Kapha differentiating in hypoxia axis. So one of my colleagues, he said that if that is so, then if I tune this axis with a known inhibitor, will there be some consequence? So he had an asthma model and he tuned this hypoxia. He found that the mouse started to have lots of inflammation, so much so that it was steroid resistant also. So he, for the first time, could show that if you actually have hypoxia in asthma, then that could have very drastic consequences. Now that would be the cause. So now what we had a model of asthmatic mouse in which Pitta Kapha perturbation has happened. So we thought that if we take a drug, which in Ayurveda is used to treat Pitta Kapha perturbation and in asthmatic condition, will that actually have a consequence on hypoxia? So we took Adatoda vasica, a drug which is bromhexane and all are some medicines which have come out of it, and we gave it to the mouse, and we found that all the features which were actually shown to be elevated by having a low oxygen condition in asthma got reversed to the extent that even the steroid resistance was reversed. And this we could find that the mechanism was it could restore mitochondrial function and mitochondrial shape, and because of which this drug was acting. Now this was very interesting because during COVID time, hypoxia was a major problem, and we decided to look up models that if it works in asthma, does it work in other conditions? And we were able to actually show that all the preclinical condition in which Adatoda vasica is proposed, we could actually attenuate those. So that actually tells us that if this evidence is created or is there in place and we have a molecular language, then we can start to translate all these knowledge. And maybe if an Ayurveda doctor is recommending this medicine in COVID time, Adatoda vasica was one of the recommendations, not because of our result, but they had recommended it out of their own yukti. And when we actually found that, they could actually take it up, and this is undergoing some trial, meaning some trials have happened in those conditions where hypoxia is compromised. So you can now think about that how this kind of thing is really, really contemporary, that you have a way to classify people, you have a way to predict who are going to have a hypoxia consequence and who are the people, what are the kind of medicines one can actually use to alleviate it. These are all based on these observations by which we are able to connect these sciences. Have 2 minutes or how many minutes? 2 minutes, okay. So, I will skip this story just to say that we've also been able to make frameworks by which if there is a medicine and if that is used for a particular usage in Ayurveda, can we use genomics framework to repurpose or to identify how that works. And one such medicine we took up, which was called as Cisamplas parera. It's used for women-related hormonal problems and bleeding-related problems, and it is also used for dengue-like condition. And we did lots of experiments. I'm not getting into the experiments. There are papers which we have published, but we could actually show that 90% of the SARS-CoV-2 titer could actually go down when we use this medicine. And this was done by molecular experiments based on usage, based on genomics experiment. We could arrive at that, which again told, and this time we found that Ayurveda doctors had not recommended this medicine, but then we went back to them and asked them, is this documented? And it was found that Patha, which is what this medicine is called, is actually been recommended in those conditions which Ayurveda has issues. So now coming to solving the jigsaw puzzle, now you can know that all these conditions which I told you, severe sweating, acne, all unrelated to me. But as I have grown with the subject, and if I talk to an Ayurveda doctor, they will say that these are all manifestations of pith in different ages. Okay, so you have at every age a way to assess why you are getting what you are getting, because you are in a Vata age, Pith age, how you started. And maybe it might be telling me that in the last part of my age, if I have a integrative approach and if I understand through a platform both these things, I might be able to have more options because I can keep my pith in check by food, by drugs, or by maybe some kind of a lifestyle change also. So this would be the true for everybody, and that is what we anticipate, that if we are able to build such kind of platform, we might have a much better P5 medicine platform, which could also be powered by artificial intelligence, because we would be capturing all this data plus the Prakriti related data. And that's what I visualize, that instead of having the two systems thinking that they are diverse, if we can get the best of both worlds with objectivity and with method of science, we might be able to have an integrative medicine. And that's what we are doing at IIT. Now we are taking these learnings and we are integrating it with engineering sciences to build ontologies and to be able to ask how we can get them to one platform. For example, how we can use ontologies to bridge what Ayurveda sees in a patient and what modern medicine sees in a patient, and whether we can break it into Vata, Pitta and Kapha by the explanation of Ayurveda, and bleeding, meaning some other description by way of modern medicine, and bridge the ontologies. So some such activities we are doing, and this is the last slide, that now we are also getting the Ayurveda people and the modern medicine people engaged by doing thinkathons, by having integrative medicine in AIMS Jodhpur, where people are now exchanging notes and trying to develop a protocol by which we would be able to get this. So this has been done by a huge team of investigators from IGIB and from IIT, a very transdisciplinary team, engineering, doctors, public health, lots of people. If they had not been there, and more importantly, Bhavna Prashar, who was there for 20 years in IGIB. Now I'm building another team in IIT, and thank you so much for patient hearing. I don't have time for questions, right?
Yeah, thank you for this talk. We are a bit over time, but let's take a couple of brief questions and then we can take more questions at the end of the session. So just a couple of very quick questions. The first question is from Murli, who sent it in on Twitter. Can you comment on the impact of understanding microbiome in managing illness, wellness, and lifestyle?
So we have done metagenomics of Prakriti types in people, because that's one of the very interesting way Ayurveda treats diseases. Microbiome is coming up important big time by metagenomics in modern studies. But now what we are doing is we are looking at Ayurveda therapies and Prakriti people and what are their microbiomes in health and in after therapeutic condition. And we have got some publications which have told that different Prakriti people have different enrichment of microbiomes, which could also tell something about their susceptibility.
Let's quickly take one more question. This was also raised on Twitter by Ravi. Is the definition of Vat, Kaph and Pith standardized? Aren't the ideas different in different places?
No, absolutely standardized. They follow the same Sanskrit text, and the standardized in terms of today's modern language, not much is done. So that is what we are trying to build objectivity. But in terms of Ayurveda doctor, they have pretty much consensus from any part of the country.
Thank you very much. There are a couple of other questions, but maybe in the interest of time, we will come back to these later if time permits.
Thank you so much.
Thank you so much.